I'm Phil Hewitt. I'm from Merck in Germany. So I'm not going to say too much. I think the best thing is to get straight on with this. And we did have a lot of discussions in the organizing committee about what a debate should be at such a meeting, because I think everyone is very like-minded here. So it's very difficult to find a topic which is black and white. So we agreed on this topic, so many choices, what models are best, but that's more or less saying, where does MPS fit between standard in vitro and animal studies? And I think we have four speakers today instead of the usual two. So I think it might be more of a discussion rather than a strong debate. So we have four wonderful speakers, so Ivan, sorry, I always say it wrong, Lorna, Annie, and Jason. So I won't say anything more apart from hand straight over to Ivan, who will start us off. And maybe introduce yourself as well.
Great, thank you. Thanks, Phil, for having me, and thank you all for coming in person. I'm Ivan Rustin. I'm at Texas A&M University. I'm a professor, and I'm chairing a regulatory science and toxicology training program there. And as Phil said, we do have quite a bit of agreement on a lot of topics. And so what I would want to do to start us off is to perhaps stir things up a little and to bring up some of the discussions that I'm hearing a lot in these types of meetings, being a toxicologist, coming to these meetings, and people say, well, animals have failed us. So I wanted to start with maybe some considerations as to where that comes from. And as I was looking for facts rather than fiction, I actually wanted to find some supporting information. So there are four statements here. They're direct quotes. They are kind of a transition point from research translation or from the laboratory to the clinic is hard to perhaps animals and are not useful. So the facts are this paper in 2003. These folks have done a literature search of the best journals and papers who made explicit claims that their findings are translatable to humans. And then they look 20 years later as to what happened to those. So it's an interesting thought experiment. And what they found is that about 18% of things that were claimed in Science, Nature, JCI, and likes that would be translatable actually only translated. And it didn't really matter what model they were from. Were they human model, animal model, molecular, and cellular model. And so that paper was cited in 2014 and kind of lamenting the fact that it's hard to do translation. And then people for ethical treatment of animals picked that up and said that animals failed us. And they used the same statistics and they cited the same papers. And unfortunately, to me, the top cite comes from NCAT's press release, which wasn't referenced to anything, but basically uses the same arguments that animal research has failed us in the clinic. So is it fact or is it fiction? And I'll come back to this at the end, but basically I wanted to point out that misleading statements like the two on top are counterproductive to research translation. So the utility of preclinical animal data and drug development. So if you ask folks who are here on the podium and many in the room, related to pharmaceutical companies one way or the other, there are 3 papers cited there, and each one of those papers says that there's great value in animal experimentation for developing of safe drugs. And then if you look on the other side, and this is a paper from British Union on the abolition of vivisection, so obviously you understand where these people stand. And in their paper, they do say that they don't believe that animal experimentation was actually useful, but they do acknowledge, and these are again direct quotes, that a positive signal in an animal study actually is quite valuable to human risk. So let's talk about purpose, right? So again, on the here, joined by me, all people to whom the chemicals are drugs. And I'm a toxicologist, and to me, chemicals are much more than that. They're agrochemicals, they're industrial chemicals, they're pesticides. And while we're taking drugs by milligram or smaller amounts, we're actually using all those other chemicals by metric tons, and we buy them actually in ounces. Now, for most people in these meetings, FDA is the only regulatory agency, but because there are many more chemicals than drugs, there are a lot of other agencies, and purpose for them will be very different. And I invite you to, again, all learn about that, and there's a great paper that summarizes all of this. Now, let's talk about 2 purposes. So one purpose is, again, these environmental compounds like pesticides, and this is a list of tests that are required for registering a pesticide. And if you look at some of them, they're like acute toxicity tests for particular toxicity. They're called six-pack. There are already a lot of in vitro and in silico models that are developed that are replacing them, and none of them, to my knowledge, are MPS. Now you go down the list and there are studies that take days or months and you are really not sure what you're looking for. So when we open an animal, we examine 45 tissues or so. So try to think about 45 tissue chips, all connected and over 90 days. Now you go further into cancer studies and those are usually years. And again, we're not sure what we're looking for. And I also wanted to acknowledge that a lot of models on this list are actually in silico or in vitro models already. So it's not like the only tests are there for animals. Now for FDA, if you, again, are trying to have a purpose as a drug, then you need to think whether it's good for your investigational new drug application, which is before clinical trials, and you need to prove to FDA that the drug is safe. And then in clinical trials, you need to prove that it's both safe and effective and intended for purpose. So there are a lot of, again, on this table, assays and phenotypes that need to be assayed. And some of these can be done by microphysiological systems, safety pharmacology, for example, but many others are not. And again, this is where another 10% argument can be obtained. Once you're in clinical trial, 90% of clinical trials fail, but largely for reasons that are unrelated to the safety package that went in. It's for actual clinical trial purpose and your population of individuals. So I wanted to conclude, and again, I hope looking forward to the conversation, that we all should celebrate, those of you who are in this room developing microphysiological systems, because they are definitely very useful, potentially useful models. But we also need to make sure that we have models of different level of biological organization that includes humans, but it also includes other organisms as well. All models are wrong, including in MPS and in vitro, and many of them fail. So we really need to do a better job in research translation. But I want to make sure that you understand where I stand in this conversation is that animals will remain in use to assure safety of drugs and other chemicals because they're required and because they are actually irrefutable evidence that they actually save human lives. And finally, I think we need to meet in the middle, and I'm looking forward to this conversation. And as a toxicologist, I need to do a better job educating my colleagues in microphysiological systems as to what is needed. But also we need to stop, I think, making some ridiculous claims about animals are wrong and MPS are best. So thank you.
Okay. Thanks, Ivan. I don't need to move. I think next I'd like to introduce Lorna from Emulate. I think has a very, not a very different, maybe a different perspective.
Thank you. Am I driving the slides? Yeah. Okay. So as we say, the topic of today's discussion is about how many choices we have to use to pick the best model for the purpose we want to use. And my disclaimer here is that, of course, my background is drug discovery and development. So again, this conversation from my perspective will be very much taken from a drug discovery perspective. Okay, so what we in drug discovery are trying to do, of course, is predict human response to drugs. And I'm glad Yvonne actually showed the figure of 90% failure rate, something him and I have talked about a number of times. And I think it is very fair to say that clinical attrition is particularly high. However, this is often an aggregate score. And what we really need to think about is the progression of good candidates into phase 1. Because if you don't get a good candidate into phase 1, the data consistently tells you that the probability of approval is only around 10%. So later stages in clinical trial tend to be, for sure, more predictive and greater chance of success. But what we're focused on at Emulate is actually understanding what are the challenges towards getting those better drug candidates into phase 1. And for us, we believe this is about having good human relevant models. Now, another point that Yvonne did touch on, and certainly an area that we've had experience about as well, is that species-specific toxicity really does play a big role in how drugs are developed. Now, I am a trained in vivo pharmacologist. I, for one, would definitely say that animals do provide value in the drug discovery process, but there are very many documented cases where animals actually haven't provided us that safety assurance that we were looking for. And thiouridine is often cited as a good example of this, and for the chemists in the audience, they'll recognize that this particular molecule is really a mimetic of adenosine. And what happened was it was being developed for hepatitis. It went through the clinical trial, through the animal models, and no effect was seen. But unfortunately, 5 out of 15 clinical trial participants were unfortunately died as a consequence of taking this drug. Subsequently, it was found that because it's a mimetic of adenosine, it interacts with the ENT2 transporter, and there are species differences there. And that was largely responsible for why this drug does cause steatosis in human, but not in animals. And we actually showed in a publication in 2019 that actually rat chips, so rat model, would also have failed us at this point. So if we are using rat chips early in the discovery process, one does still have to be mindful of species differences. Now, we've heard a lot in this meeting about interesting science, and we've heard several people, Don Ingbert and Mike Schuler again this morning, Jay Hickman yesterday, talking about the fact that actually our technology, microphysiological systems, is actually coming of age, and it is moving from just interesting science to IND submission. And I think, again, I want to bring us back to this concept that we're focusing on building models that actually get better candidates into phase 1. So with that in mind, what we're looking at predominantly, and my guidance perhaps and conversation I'd like to have with this team here, is that picking the right model and building the confidence that human relevant models will drive better candidate progression relies on, first of all, developing a good model system. There are guidelines out there. I think we could benefit from more guidelines. Several of you have actually mentioned that to me during the course of this particular meeting. Experimental design is absolutely critical. Understanding how many drugs to test the model system on, how many donors, perhaps to look for reproducibility. Important, I'm a pharmacologist, so adjustment for protein binding is also essential. And good hygiene in terms of blinding scientists when they're preparing these data sets are, again, going to be key to developing good models. And finally, once we have got those two things, we need to, of course, evaluate the performance. And many of you know that we've been doing this in Emulate. It is on bioarchives. We're focusing on the liver. We've looked at 27 small molecule drugs now across 3 donors. And repeatedly, we see a very high sensitivity in the ability of our model to correctly identify hepatotoxic drugs that have been in the clinics. This is a retrospective analysis. And because we know they've been in the clinic, we can say that if the animal model did detect a hepatotoxicity, it was certainly not used in the decision-making to prevent that drug from going into the clinic. So here we show the performance of these types of models, and of course more would need to be done. So lastly, I've talked about progression of molecules into phase 1, and where I would advocate then a model such as ours would actually reside. And we firmly believe that this model resides in the lead optimization process, and importantly should coexist with the animal model. So I think that might surprise some people in this room. I'm going to say that for the time being, these models can coexist. But we believe that if you place this before your early dose range finding studies, there is the potential for us to start addressing whether animals are actually even needed. If we see a toxicity in our liver chip, then our recommendation is that is a real stop and think moment. In summary then, if you've developed the right model, you understand its context of use and you're using it to progress molecules into phase 1, we do believe that this will cause a reduction in the failure rate we see in the clinic. Thank you very much.
Yeah, so Lorna, thank you so much, and I appreciate the perspective and the commercial for Emulate as well, right? So who's probably a sponsor, right? So briefly, I think what we need to really think about is the cost and the need and the volume of testing that needs to be done. And I think it's totally fine to have fantastic systems that can be inserted surgically in a particular point in either drug safety development or in any other safety paradigm. But to kid ourselves that this beautiful science that all of you and us are doing is going to replace animals, I think is preposterous. And I hope that we have a conversation about that.
just adjust the microphone down. So, thank you, Yvonne, for those comments. I won't directly rebut those at the moment, but I will rebut some items I saw in your presentation. So you talked about misleading statements for translation, and I do actually sadly agree with you on that point. And I think we should have a bigger discussion about how one uses the data that's available to us. So, for example, folks sitting in the audience that are from the pharma industry know that actually some of this data isn't published on a regular basis. It's quite out of date. It doesn't reflect therapeutic indications, nor does it reflect therapeutic classes. So I think if we could get greater granularity on that data, I think it would certainly help us understand whether our statements are really true or genuinely misleading. My other comment really to you as well is that we're all learning here, and I know that I've seen John Wicks go somewhere in the audience. I saw him earlier, he's got a beautiful pink jacket on, but I can't see him right now. And so, John, I am going to steal one of your quotes. So you talked about models being wrong, and actually I do again agree with you on that point. A model is a model, and John has a beautiful way of describing this, which goes along the lines of, if you want to model a cat, you need a cat, preferably that cat. So I shall leave you with that and pass over to Annie.
Okay, good. I got it now. So I'm Annie Moisin, Program Director for HOPE, so Human Organs, Physiology and Engineering at Wellcome Leap. Prior to that, I was in mechanistic safety at Roche for many years, and developing MPS and also running standard toxicology assays. So from both angles in my work. So thank you, Ivan and Lorna, for the evidence, the numbers, and so on. I think I'll be the simple-minded one here, perhaps, and bring this question down to fundamental biology. Species are different. And it's a fact that species-specific expression level of even a single molecule on a given cell type can lead to dramatically different body responses. So illustrated here, the blue cell type you see was activated by an investigational new drug in human, and it wasn't activated in the monkey. Consequence, catastrophic organ failure in 6 men at a dose 500 times lower in men than in the non-human primate before. So I'm referring, of course, to the infamous TGenero trial. So in that case, the CD28 superagonist antibody activated Tregs and promoted tolerance in animals, switched on T-cell helper T-cells in humans, so triggering a cytokine storm. And this is not just one anecdote, so we've seen more of that. Do I switch slide? Okay, yes, yeah, and so let's be a bit more forward-looking now. So there are more biologics to come, not only in the pharma industry, but also in agrochemicals. And generally speaking, and more biologics then will mean more species-specific or human-specific reactions. Generally speaking, you all know that compared to small molecules, chemicals, biologics are most of the time more target specific or target specific to their target sequences, and often they don't even cross-react with animals. And that poses a great challenge, of course, for preclinical development if we don't have good human-based models around. And second, unlike small molecules, biologics are bigger, highly engineered, and often they are recognized as foreign bodies by the immune system. What happens? There's a T-cell dependent antibody response. The antibodies can alter the PK, the PD of the drug, compromise efficacy, compromise safety, and this is very poorly predicted with animal models. So we need human models for that. I'm not an expert, certainly not in agrochemicals, but seeing the growth rate of biologicals, and most of them, I think 50% being microorganisms, microbiomes, I really wonder depending on the exposure, of course. What's the risk for our microbiomes? And microbiome, that's also an area where, forget about animal models there, we need human systems. So we've learned for many years that in terms of immune biology, mice and humans are very different. And we are learning now with more studies, and there's a recent one from 2022 that we are also quite far away from monkeys. We do have similarities, of course, but if you look at level of divergence, and now we are learning about cell identity, cell type frequencies, expression level of key receptor for drug developments, just some examples here, MHC, FC gamma receptor, the STAT pathway, interleukin, and so on. I think there are sufficient evidence that either we start also humanizing non-human primates like we do with mouse, because if we want mouse to be predictive, we humanize them, right? So shall we do that? I don't think so. I think we should just go ahead with human-based systems. And so we can use blood, human blood, that has some limitation, of course. We can look at acute responses with fresh blood, some cytokine release, for example. But you've probably, you heard that also this morning in the session, immunity occurs in tissue. That's where antigens are sampled. That's where immune cells crosstalk and they meet and they mount the adaptive and innate immunity over time. So what we need, if we want to recapitulate the human immune response and then be more predictive, we need to create this tissue microenvironment, seeing immune cells being infiltrated, recruited, and responding over time. So we need longevity. And the good news is MPS can actually do that. I think we have sufficient demonstrations from yesterday, this morning as well. We'll see more pointing towards feasibility. We thought it was impossible. I think it's getting really possible to do. And I think it's not really scientifically sound nor ethical to try, fully humanize a mouse or a monkey. It's not an animal anymore. So let's just like push forward and bring MPS to the next level. And the time is right to do that, because now we can read tissues thanks to spatial multiomics, single cell analysis, like never before. So we can extract the information we need as guiding principle for engineering these tissues. It requires resources and tenacity, of course, but I really do believe that MPS is the future, and I hope we have the courage to address the human relevant question with complex models like MPS.
Thanks, Annie. Final speaker will be Jason.
Yeah, hi everyone. Yeah, Jason Eckert. I lead the complex in vitro model group at GSK. This will be my opinions and thoughts on this area. Okay, perfect. So where I stand, I think it depends. So when I say it depends, it comes back to the context of use. It has to be dependent on what you're looking to do with a system or a animal. And I put this up here. This links with some of what Ivan was talking about. So on the right here, looking at clinical experiences, and this was a recent study in the biopharm space, looking at how our failure rate, which in the drug discovery is quite poor. And then on the left here, where I'm talking about from thinking of the context of use, and this is from a recent paper where there was a workshop with the FDA and then the IQ MPS affiliate, we put here different buckets where we see the potential for MPS in the future. So where we're hoping to push the limits of having MPS earlier in discovery, and then also bridging that gap where Lorna was saying, can we use MPS to help better predict early toxicity, and then also that rather than potentially replacing straight away, it will reduce the number of animals. And then eventually, and this, if you read this paper, we talk a lot about depending on the context of use, for example, in the safety or ADME space, it's going to be a weight of evidence where there's going to be an increasing amount of MPS data and that will support the animal modeling as well. So where I wanted to show two different areas where, from a context of use, and this also links with quite nicely with Annie's comments about biologics and also looking at cell therapies, is on the right here, there's a plethora of different models where GSK has been working on in the oncology space. So from tumor spheroids, which have been around for decades, to more recently using tumor organoids, which are a static system, where we've been using tumor organoids to look at drug-drug interactions. So that's moving closer to the patient, where we can look at these drug-drug interactions rather than waiting to go into the clinic. And then that can help determine what potential animal models we could use. So where we could do a organoid model, and then you could actually do a PDXO model in an animal. So you still potentially may need an animal before then increasing your confidence of then going into the clinic. And then as you see going down the diagram, there's increasing complexity of these models, where you'll have a multi-layered system where we've been using a triculture system, and this was looking at PARP inhibitors, and then vascular models where we've been using models where you have tumor cells embedded with a vascular system, and also where we've been looking at movement of T cells across endothelial barriers on the right here. So it depends, as I said, on the context of use. And then you get right to the bottom here, where you can have microfluidic models that have an ex vivo tumor tissue, and this will be even closer to the clinic. And I have on the left here, and I've crossed it out, you can't see it, hiding behind that X is the animal. And I think eventually that's where we want to be, but we're not there yet. And then just one switching gears, and this links with what Lorna was talking about from a liver in vitro models. We've been developing a number of different liver models within GSK, and from where we've had in the past using the sandwich culture systems. We've used liver spheroids, and we've used the liver spheroids quite successfully looking at early hepatotoxicity. And so this has helped drive using a battery of different assays along with the liver spheroid models to better predict which models are going to have that hepatotoxicity. And so therefore we will stop the progression of those molecules depending on that battery of assays. And then on the far right, you've got different liver on a chip models, and we've been using these to look at different diseases such as NASH, and you could use it for in the infectious space for HBV. And so for those models, you have to think about from a cellular context and cellular architecture of what cells you would need when developing that model. So from my point of view, we're moving in the right direction, but we're not quite there yet. Thank you.
I see Ivan has his hand up, so yeah, I mean, I just want to thank all of you first before we carry on, so I don't have too much to say on top of that, but I want to also encourage people to ask questions now. I know Ivan has a rebuttal or a comment, but please, what is your opinion? What do you think? Are there people in here that believe we don't need animals? So please be active, but over to you first, Ivan.
Well, thank you all again for speaking up your mind. And as Phil started at the beginning, we really do not disagree, right? And this is not a debate about animals, right? This is really, I think, a conversation about where we go, where we are, and are we there yet, or how soon will it take for us to get there? So I certainly don't think that in my lifetime we'll be anywhere with, emulate. The only thing that is on the drug development path is emulate chips, or any other chips for that matter. But I think I wanted to come back to a couple of points that Annie made, that MPS is the future. I think for certain things, absolutely, that they will be used as was shown in drug development in certain places, absolutely. And I think we all need to develop basic research and move this forward. However, we need not to forget about the reality, and the realities are challenging. Realities are challenging in terms of cost. Realities are challenging in terms of throughput. Realities are most challenging about the cell availability. Realities are challenging about reproducibility and how many experiments you need to run to really, have statistical power to make any conclusions. And to do that, I think the groupthink need to occur. And I think the developers and the users need to, again, do a better job educating each other about what the needs are and what the challenges are. And that's what we're hopefully going to have a conversation about today.
Anybody else? Are there any questions? Please go ahead.
Yeah, when I saw the title of this, I thought we were going to actually be talking about different MPS models rather than whether or not animals will be part of the pipeline. So I wanted to maybe see if the panel might think about the topic that when I see the vast number of different MPS models that are being developed, it seems so much larger than the number of animal models that we have: mice, non-human primates, rabbits. And so how do we think about the hundreds and hundreds of different MPS models that are being developed? How are they going to sort of fit into the pipeline? I'm not quite sure I know the answer to that. I would be interested to see what you guys think.
Yeah, I can take a stab at it first. It also will be dependent on if it's fit for purpose or context of use. If it's in the safety space, I think where there's discussions through the IQ MPS affiliate and with regulators is that we need certain standards to start off with. And then with those standards, depending on what your specific use case is, that you then may have to have further standards or certain criteria that you have to set up.
Maybe just a quick comment. We tend to think that we have mice, rats, non-human primates. These are preclinical models, but think of all the strains, that you can choose. You really choose also your mouse specifically for the question you want to address, and often you keep changing the strains until you get the answer that you really want. That's true. So there's quite a range of I think animal models as well, very wide range here. Yeah, just a quick comment.
And I actually think it was an excellent question. Thank you for asking it. And I think one thing I would bring us back to is folks who sat through Mike Schuler's presentation this morning had a really nice history lesson on where a lot of these concepts started. But I think most people in the room would agree that while there are good examples of these types of models being developed 20, maybe 25 years ago, the whole field's really come of age in the last 10 years or so. So I think at the moment, yes, you are seeing lots of different ways. The example on the slide there is great, shows lots of different availability of liver chips, I think, with standardization. And I think as we really put those models through their paces, what we'll find is that there'll be some models that are better, and we can discuss what we mean by better, of course. And those will be the survivors, if you will, as we go forward. So I see it maybe a wee bit differently from yourself, and I don't think we're going to advocate for having 10 liver MPS models in the drug discovery process, for example, but I think through time we will eventually see one or two that are more superior and to Jason's point, fit the context of use.
I also had one of my questions would have been on that similar line, that we have a lot of models, we've heard about a lot of them, but one of your first slides, Ivan, in talks, we need to look at, well, you mentioned 45, but I guess normally 30 something organs. We don't have enough models for most of those systems. So we're a long way from reality, from talks, to actually replace an animal.
But also, I think what we need to think about is what does success look like? And to many developers of new models, success only is true if you're replacing something or if you knock something off its existing position. And it's one way to look at it. But I think what we're seeing in biology in general, and biomedical engineering is not the only field that is undergoing tremendous expansion, is that it's okay for scientists to try different things, to try hundreds, thousands of different models. But scientists at the same time need to understand what the utility of those models will be to increase the chance of creating something that will be translatable. Because as I've shown you in one of my first slides, just declaring that something has great promise really is very opportunistic. So if we lower the temperature, and if the success doesn't necessarily look like FDA now starts accepting these data, or model X replaces model Y, then we can all coexist and all walk together to the future, which again, hopefully I retire by then.
Thomas?
So I really like the role of an agent provocateur, which Ivan did take here. And I'm so relieved that all the others which have background from pharmaceutical industry were so much more positive about MPS, because that's what is really driving their use. I think we should, the problem we are doing is any type of black and white. To say it is either animal or MPS is simply not realistic. If you're spending upwards of 3 billion for getting a successful drug to the market, every model which will change the odds of getting something faster or better out will be used and should be used because it is increasing safety. But I think we should also not say it is an abuse of data, then cite Olson paper of 2000, which shows that rodents only found 43% of the adverse effects of drugs. The Olson paper was praised for how well animal models helped us. For most substances which are not drugs, we only have rodent data, and 43% is simply not enough for me. I would really like to have systems which are finding more of these, and at the same time, I think we're also killing people because we're killing substances with wrong animal data. And this is not only an animal problem. I don't want to know what the genotox test battery has done to drug candidates, because we are sorting these out in the development process with something which typically more than 90% does not hold as soon as you go into the animal. So we have a problem, and this problem we can only solve by more cleverly integrating the various approaches. One of my most recent papers I have entitled "A Call for In Vivitrosi," in vivo, in vitro, and in silico combined, and that's actually where I see the future. And MPS somewhere is in the abbreviation as well, but I think it's a driving force for making this actually a very successful mix. So one was no question, but a comment from my side.
Can I start with it? Thomas, thank you so much. And again, I'll have to reread the Olson paper to fact check the 43%. But I wonder, what is the alternative? You know, we all talk about, you know, a case example of one clinical trial, you know, killing 6 people. Thalidomide is a favorite for animal abolitionists in toxicology. Those case examples do exist, and no model is perfect. You know, engineers are great at engineering best things. Cars still break down, rockets blow up. Things will fail for reasons that are uncontrollable by us. And what I am concerned is that we need to treat people type of argument. We need to have better data on people, that we're actually talking about either not testing anything and throwing it straight into people, or testing it on MPS or any other in vitro system and then going straight to people. I am concerned that people will become test species for anything, drugs or chemicals, without any filter before. And I think, unfortunately, we're seeing the practice of this with cosmetics. Cosmetics cannot be tested in animals anymore. And yeah, I don't care about cosmetics. There's enough perfumes and ingredients and other things. But if we're going with politicians pushing us the same way on agricultural chemicals and industrial chemicals on drugs, I have a real problem with that. I don't want to be the test for something that could have been tested in other models, including MPS.
So Ivan, I'm going to rebut you on that in relation to some of the comments you also made regarding the work that we're doing. And I think you did use a word that I use, so I take that as a compliment, which is coexistence. And I think what Thomas is saying, what I'm saying, and other people in the room will say, is that we're not saying today we're ready to replace the animal and we'll remove the animal. I think we actually have to go through a process of learning about these models. And that was why I was advocating the approach that I align myself to now is thinking about good experimental design and the pursuit of data that will really allow us to understand whether these models are part of what I would term that integrated risk assessment. Now, I take my inspiration from the work that's been done in the cardiovascular safety pharmacology field. So again, we can go back to the 1980s when drugs were withdrawn because of the torsades. And it was several years later that the molecular entity, the hERG channel, was identified. It was 10 years after the identification of the hERG channel that there was then some regulatory guidance for the inclusion of a GLP hERG study. If I bring that right up to date, that community have worked hand in hand with the regulators, some of whom may be in the room, and are now saying that it's an integrated risk assessment with a hERG, with the dog in vivo cardiovascular study, with the CEPA initiative, looking at the other other ion channels, I beg your pardon, that drive the action potential, and importantly still a clinical thorough QT study. Okay, and it's when you see negatives across all of those that's what drives the confidence that that is safe to progress into humans. And I would like us to use that as our inspiration of when we're building our new models. We focus a lot at the moment on safety, but remember these models will be good for efficacy as well. They will help us in areas beyond drugs, but I think we should use that learning and how we can maybe shortcut some of that time process that it took from the 1980s, where we discovered torsades, to today, where we're now maybe getting into a place where regulators and pharma can coexist. So for me, coexistence is the key.
Yeah, just one additional point to Thomas's. I think it's a great initiative or thought of where I know GSK is probably not the only pharma company where we're talking about in silico modeling group, both in the safety, so looking at potentially how you can incorporate into adverse outcome pathways, and then also from a PK or PD point of view, when we're modeling for dose prediction, we're speaking with them as well. And also looking at from a quantitative systems pharmacology approach to understand what cellular components do we want to have when modeling our MPS models.
I want to follow up a little bit on what Thomas mentioned at the end of his question. And I very much appreciate the discussion on these models, but we are talking experimental models here, and these models are not run in a vacuum. It seems to me we're missing a big component, which you just kind of mentioned about modeling, which is the computational analytics end of this, which is, as we know, going forward into omics type analysis is becoming more and more not only important, but critical, key components. So I'm thinking that these MPS models, or maybe even animal models, can't be run as a standalone answer anymore. That has to be integrated with a more and more sophisticated computational approaches. Maybe you can address that. Thank you.
Well, I wonder if I can, Larry, maybe expand on that and ask my colleagues who've been at pharma or are at pharma, did the recent example of omics, did that really, speed things up, lower the cost, improved safety profiles, realistically? It's been 15 years of genomics, right?
I'm sure you'll get a different answer from every farmer. I know at Merck, I would say no. But I would also argue that after those 15 years, now it's a standard tool where you can take it out of the toolbox and you will run it. And I do agree myself that all the data that's been generated in these systems will rely on some sort of, I hate the word AI because you use that for everything these days, but that has to be an integral component to understand all this stuff. It will never go anywhere.
Yeah, and I know that I've spoken with a couple of people here. For me, the modelling side of things actually will help drive the improvement in some of the wet biology models. So less about the omics, Yvonne, but one thing that I'm particularly interested in is how we can use mathematical modelling to help us understand the right model to build. And so I was having a conversation with someone earlier about the hepatocyte. If I produce a hepatocyte in my liver today, it will be in my liver for the next 9 months. And, what we have to ask ourselves is, do we therefore need experimental models, in vitro MPS type models that last for 9 months? And I don't know if we can actually genuinely answer that question. I don't know if Adrian's in the room, but Adrian for many years ago had a nice slide up that they were culturing hepatocytes over time. And what they found was that those hepatocytes became less hepatocyte-like during the culture period. And so I'd like us to use mathematical modeling to help us understand the kinetics of the biological response and use that in a good way to design the right model. And ask ourselves the question, do we really need 9 months of hepatocyte viability in a model system to allow us to really replicate what's happening in vivo? So I think there is multiple areas where mathematical modeling and AI type approaches can actually help enhance what we're all trying to do.
And also to the omics, omics was mentioned, I think, in the identification of better biomarkers. That's what we need to do, also to have a non-biased integrated approach to biomarkers, because we are constantly looking for biomarkers that are predictive of responses, and it cannot be a single biomarker giving us the answer. So just going like fresh reset and then look at the data with the new technologies that are available now.
Conceptually, this is great, but I'm not sure how many of us have looked at the bottom line and the budgets that we have. So conceptually, throwing everything and integrating, right? It's fantastic, but the reality bites, unfortunately, and I think that's should be something that everyone considers as well. Is what is the most useful model for the purpose that I have, and regardless of how much beautiful stuff is out there, we probably will not use 99% of it.
So I would say over this.
So this debate about what model is best and what model is most purposeful, I think it can be settled at least partially if we can come with some ground rules as to how we are going to validate the model. Now, if you're making a microphysiological system and you want to go and publish this in JCI or any other traditional high impact journal, for example, or you're putting a grant, it's almost very common that there is going to be at least one reviewer who's going to ask you to validate your model with a mouse model. Now, you can do an entire study with the mouse model and go publish in Nature, and there's going to be no reviewer who's going to be asking you to validate a mouse model. So this dogma that a mouse model is physiological has to be broken. Now, that I think is more preposterous than anything, that even though we know that first slide that you showed, Ivan, that mouse model is considered physiological. I think that should be part of this conversation as to how we can inform the community and how we can break this dogma. Only then other experimental models will thrive. And it is particularly important for early career and mid-career scientists, because if they want to grow and they want to build this field, then they really need this space where they're not tortured by this idea that they need to validate their models with the mouse model.
You're preaching to the converted.
I like the comment very much, and I think we need to bridge worlds, like the academic worlds, the pharma, and also regulators, because what happens when you submit a manuscript, it's going to be reviewed more by academic labs, PIs, and some lack of awareness of how we actually back translate and validate the models using preclinical data, using clinical data. And why? Because this is also not public. So there's a problem maybe also of sharing approaches. So how in pharma do we think a validation should work? And then what is happening in academic labs? That's really two, unfortunately, often separate worlds. So bringing these communities together first and then raise the awarenesses. Doesn't make sense to validate or to compare to the mouse if I'm developing a human model. We need to improve that.
So I appreciate this comment about reviewer number 3, because exactly the same argument I've heard in a couple of different meetings in the last year, that the reviewer asked us to compare this to some, in vivo model, and this was the wrong question. I don't think it's a validation question, it's the relevance question. There's nothing wrong with asking, how does your model, whether it's on flat plastic or 3D or something else, how does it replicate something real? And perhaps reviewer 3 was inartful in explaining that, but to me, it's not a validation question, it's a relevance question. And relevance is a very important scientific concept.
So Ivan, maybe a question back to you about, I know the flip side could be comparison to a 2D. A lot of the work that you did in for the validation center was the 2D. Was there thoughts of having a different comparator?
So it depends on, so Jason mentioned, we have a Texas A&M tissue chip testing consortium, which originally was started by funding from NCATS, and now we have 8 members who contribute each year, and we do experiments together to try different things. So in our point, you have to have some sort of an anchor. And if you have an anchor to human physiology or to animal physiology, you'll be benchmarking to that. But frequently the anchor or the benchmark is the status quo or standard of care, right? Just like in drug development, you're always or almost always comparing your drug to the standard of care. The similar concept applies in MPS testing. If we would have a particular MPS replicate a particular human condition, and if we would have enough replicates or to run multiple drugs, perhaps you can do that. But in absence of throughput and in presence of very high cost and complexity, you can only compare to 96-384 well plate, which everybody does and makes decisions. And we're here today because of those models.
I'm aware of the time, but
hi, I'm Chris Boyer from Tessara Therapeutics. I'd like to pick up on the question of which specific MPS model would choose. And Lorna, you talked about a shakeout that is likely to occur. You also talked about what factors determine the best model. And we can think about reproducibility, we can think about cost, physiological relevance, other factors, but certainly one of them is the reference data set behind the particular model. And that seems to be one that's cumulative and sort of self-reinforcing. And in the end, it becomes almost like a network effect that you want to test your compound in that model just because it has such an impressive reference set, and you can make inferences based on the other molecules that have gone through. So I'm interested in your thoughts and the panel's thoughts on how that reference data set would probably become the dominant factor determining which MPS systems go forward.
Yeah, so I'll maybe explain a little bit in detail about what we did. So we used guidance from a third party, it's the IQ MPS consortia, who actually published a manuscript in 2020 and said, if you are going to develop a liver microphysiological system and look for drug-induced liver injury, there are 20 drugs that we would recommend that you test. Now, what was in those 20 drugs, a couple of features. First of all, they represent the major mechanisms of drug-induced liver injury in the small molecule therapeutic class. Secondarily, they had all been in the clinic, so that there was, that true outcome, or if you will, there was the ground truth was what did it do in the human. But they also asked for, they gathered together 7 matched pairs where these were structural analogues, and one of them, so trovafloxacin and levofloxacin is a good example, where trovafloxacin has been shown to be far more toxic in the clinic than levofloxacin. So they were also asking for the ability of a model to start to distinguish those, and that's important because, as you know, in pharma, quite often you're rank ordering, you're trying to promote and progress the most promising candidate within a chemical series. So there was 20 drugs there, and we supplemented, when we did our study, we used 27 because we supplemented with some other ones that we were interested in that other in vitro models had failed to potentially correctly label as toxic or not. So there isn't a consensus data set. There's certainly not a consensus number of drugs, but we believe that it's one of the largest studies, single studies that's been conducted to date. And I've spoken with a number of people who have asked if we can make that data set more available to do exactly as you've said, and use that to start benchmarking own internal compounds. But if we take inspiration from outside of the pharma industry and perhaps look at some of the work that's been done by folks in the audience who were more on the chemical side, they are further forward and developing, official guidance, in some cases OECD guidance, that says these are the test drugs that one should test, and build, and show the relevance of the model to that. So I think we've still got a long way to go. I'm exceptionally proud of where we've taken our models so far, but really recognise there's still a lot more work to be done.
In the true spirit of debate, I'll point out that I think it's a false premise to try to just define your reference set, and then after that, everything will fall into place. Yes, OECD test guidelines do have that, and that's the basic benchmarking of your model, but that doesn't guarantee acceptance. The folks at the National Toxicology Program have done a yeoman's work and tried to define something as what should be very straightforward: estrogen receptor agonists and antagonists and inactives. And they really went and spent a couple of years on that. And still, it's useful to show that there is promise, but it's not going to guarantee. Pretty much every QSAR model has 75% or 80% accuracy, but the domain of applicability of any application, 20 drugs stay in the drug lane and only particular chemistry, do not go into agrochemicals or PFAS or anything like that. I think we need models that really have confidence, and that confidence will come in part from reference sets, but in part from just robust use, and robust use means throughput and cost.
Yeah, but costs mean that you need users to reduce the cost. So I think we should also not just like wait oh, but that's true. It's the wider adoption and application, and that will make the assay robust. And so we have to, at some point, also be confident and have the courage to use them and distribute them and test them and reduce the cost.
Just quickly, as a testing center, we calculate the cost and we compare different models, and the cost varies 1 or 2 orders of magnitude already. So it's not the users and then the prices will come down. Some things will never come down, and that's the very unfortunate truth.
So to add from a cost point of view, so instead of going towards a more complex model where we went at GSK to using the liver steroid model, and there's a paper, I think it's 2 or 3 years ago, by Will Proctor and others looking at, I think they had 150 compounds for looking at liver steroids. And more recently, so Chris Schofield and others at GSK have a paper looking at 170 compounds. So it's depending on how many compounds you want to look at and different classes that you'll then understand the sensitivity and specificity.
Thank you.
So, I mean, I would just add on to that you'll never have enough compounds. You ask the statistician and they'll always say they want more. But I still think it's a good place to start. I mean, the more complex the model, the fewer compounds you will be able to test you because of time and cost and resources. But it's that confidence. Once you start getting confidence, then you'll get more people testing it, then you generate more data, and then at some point, maybe in the near future, we'll be in a better way. But I'm looking at the watch, it's exactly 1:15, so I think we should stop there. I think there are no more questions in the audience. So I want to say a big thank you to all of you. It was a nice discussion, and thanks, of course, to the 4 speakers up here. Thank you very much. I just wish you all a nice afternoon and see you soon.